Okay, we are beginning this semester by looking at DNA fingerprinting, and before we do that, I thought it would be a good idea to review DNA structure, since all of the techniques that we're talking about are you're going to have some knowledge of the structure of DNA to figure out how it works. So, DNA stands for deoxyribonucleic acid. You all should have had that in biology. Its shape is a double helix. It's what makes up your chromosomes. Remember, your chromosomes contain your genes, which have your genetic characteristics. So there are two molecules that are arranged in more of a twisted ladder structure, and that's called the double helix. They are made up of building blocks of tiny subunits called nucleotides, and there are three parts to a nucleotide. There's a phosphate, there's a sugar, and there's a base. And there are four nitrogenous bases in DNA: adenine, thymine, cytosine, and guanine. So all of the DNA that makes up who you are is different combinations of these bases in different orders. So there's only four types of nucleotides: one that has an adenine on it, one that has a thymine on it, one that has a cytosine on it, and one that has a guanine on it. So that is a picture of what a nucleotide looks like. Phosphate of one nucleotide is hooked to the sugar of another, and that creates a long chain in which the nitrogenous bases are in the center. So two of those chains are held together when those nitrogenous bases bond in the center. So the phosphate and sugar forms the backbone, and the bases are the middle of that ladder, or the rung of the ladder. So here are the nucleotides with the bases on them: adenine, thymine, cytosine, and guanine. What you'll notice with the shapes of the bases is that adenine fits into thymine's shape, cytosine fits into guanine. So these bases pair in a certain way. So I know if I have an adenine, then it will automatically be bonded to a thymine. If I have a cytosine, it will automatically be bonded to a guanine. So each base will only bond to one specific other base: adenine. A bonds with T, C bonds with G. So they form a base pair, and this is called complementary pairing. Complementary pairing, excuse me. So that if I gave you one side of a DNA molecule, you could tell me what the other side is, since we know that adenine only bonds with thymine, cytosine only bonds with guanine. So the sides are the phosphates and the sugars. The inside portion are the bases. The bases are held together by a type of bond called a hydrogen bond. So notice thymine is red, adenine is green. They are always together. Guanine is that purplish pink with the green from the cytosine, that they are always together. So you will not have adenine bond to cytosine. You will not have guanine bonded to thymine. So because of this complementary base pairing, the order of the bases in one strand determines the bases in the other strand. So if you put them together, adenine goes with thymine, cytosine goes with guanine. So if you're going to build the other side, knowing the base pair rule, you would know it would have to be there we go, it's down here, but let's go back a slide, because I thought for sure that it would show you the answers on here. The other side would be T, A, T, A, G, T, A, C, G, C, C, C. So how this relates to the genetic code. When you look at DNA, you are looking at the sequence of those bases. There are certain parts of the DNA that code for proteins. So they hold the code for amino acids and give you long string of amino acids which make up your proteins. There's also part of the DNA that's considered to be junk DNA, and it does not code for anything. What we look at in forensic science is that junk code, because that junk code has repeating segments that have different numbers of repeating segments for different people. So for most of us, most of the time, what we're concerned about is the part that actually has the genetic code. And when you look at that genetic code, 3 bases on DNA codes for 1 amino acid. And that triplet of bases was called a codon. So each codon codes for 1 amino acid that makes up a protein. So a gene is a section of DNA that codes for a protein. Each gene has a unique sequence of bases, which gives the protein that it makes a unique sequence of amino acids. It will code for the production of protein, and these proteins or combinations of proteins are what give us our phenotype, our hair color, our eye color. Proteins run all your chemical reactions in your body. So if somebody has a disease, it's usually because a protein has not been made correctly. For example, sickle cell anemia, where the red blood cell is the wrong shape. There is one DNA base that is wrong in all of the thousands of bases that make up hemoglobin. It changes the shape. It no longer functions. So that order of the bases in the DNA determines the proteins in your body, which determines your phenotypes. So go ahead and see if you can answer this review. What does DNA stand for? The shape. So if you're watching this video in class, we will pause and go through it. If you all are at home, pause the video and see if you can get the answers right. Okay, I'm going to go ahead and go through the answers since we're doing the same thing in class. DNA stands for deoxyribonucleic acid. Shape of DNA is a double helix. Adenine always pairs with thymine. The sides of the DNA ladder are deoxyribose and phosphate. Guanine always pairs with cytosine. The complementary sequence to AATGCA is TTACGT. And the two sides of the DNA molecules are held together by hydrogen bonds. DNA is composed of repeating subunits called nucleotides. And the four bases that make up the rungs of the DNA are adenine, thymine, cytosine, and guanine. So that's all on the background information. There's a worksheet that you need to do on DNA structure, and we will start to talk about how we use this in forensics and what type of tests that we can do next time we meet.